All right, welcome to the deep dive. Today, we're going deep on how antimicrobial agents really work. You sent us this awesome source that basically lays out the blueprints for these, honestly, these life-saving drugs. And our goal today is to try to really understand, the core strategies, the core blueprints, if you will,
Yeah.
that make these agents work.
Yeah, absolutely. It's about understanding the different ways that these agents can attack.
Yeah.
And your source highlights four key mechanisms: interfering with cell wall synthesis, inhibiting protein synthesis, interfering with nucleic acid synthesis, and inhibiting membrane function.
Right. So it's like a four-pronged attack.
Exactly. And knowing these mechanisms, helps us to both understand how current therapies work and also how we might be able to develop new ones.
Okay, so let's just dive right into the first one: interference with cell wall synthesis.
Okay.
What is it about the cell wall that makes it such a prime target?
Well, the fascinating thing about the cell wall is that it's a structure that's really specific to microbes like bacteria, right? It's basically their outer skeleton. One of the key things here is that our cells, human cells, don't have cell walls. Right, right. So that's a really important point. And a classic example of an antimicrobial agent that targets cell wall synthesis is penicillin. It blocks the enzymes that bacteria need to actually build and maintain that wall.
So since we don't have those walls, these drugs can target the microbes without harming us.
Precisely. Yeah.
Okay?
By disrupting this process, you're basically weakening the microbe's structural integrity. It's like dismantling their suit of armor, and eventually that leads to their death. But one thing to keep in mind is that bacteria are clever, and they can develop resistance mechanisms, like producing enzymes that actually break down penicillin. So, it's a constant battle.
Oh, wow.
Yeah.
That makes total sense. So you're basically disarming them.
Yeah, exactly.
Now, moving on, going a little bit more inside the cell, it seems like equally critical to disrupt their ability to actually build the machinery they need to function.
Yes.
Which brings us to inhibition of protein synthesis.
Yes.
What's the deal there?
So protein synthesis is, absolutely fundamental to life. I mean, all living things need to produce proteins, and microbes are no different, right? They need proteins to carry out basically every single function.
Right.
So if we connect this to the bigger picture, if a microbe can't make proteins, then it can't grow, it can't metabolize, it can't replicate, it can't really do anything.
So if we can block protein production,
Yeah.
we can basically shut them down.
Exactly. And some antimicrobial agents actually target the ribosomes of microbes. Ribosomes are like the protein factories within the cell, right? And so by binding to these ribosomes, you can disrupt that protein production process. And a good example here is, drugs like tetracycline, which actually interfere with the ribosomes.
Got it. All right. So that's protein synthesis. Now we get to interference with nucleic acid synthesis.
Yeah.
And now it sounds like we're getting into like the genetic material.
Yeah, we're going even deeper, right? So nucleic acids like DNA and RNA, those are basically the blueprints, the instruction manuals, for a microbe. And they're essential for replication, for repair, for all sorts of functions.
So by basically messing with their ability to copy their DNA or even just repair damage, we can stop them from reproducing and multiplying.
Exactly. And this raises a really interesting question about specificity, right? Some drugs, for example, certain types of fluoroquinolones, they interfere with the enzymes that bacteria specifically need to replicate their DNA.
Oh,
Right. And so this prevents them from multiplying. It's like disrupting their ability to make photocopies, or to fix typos in their code. So it's really fascinating.
Wow, that's really interesting. Okay, and then finally we have inhibition of membrane function. So we talked about the outer wall, but now we're talking about the inner workings.
Exactly. So the cell membrane, it's kind of like the inner boundary, of the cell. It controls what goes in and out. It maintains this kind of delicate internal environment for the cell. And so disrupting this balance, this delicate balance, can have really, really serious consequences for the microbe.
So how do antimicrobials actually target this membrane? What do they do?
So some antimicrobial agents can actually insert themselves into the membrane and disrupt its structure, disrupt its function. It's like, poking holes in a balloon.
Oh, yeah.
And that's how you get leakage and everything starts falling apart. A good example here is polymyxins.
Okay.
They're a class of antibiotics that work this way. And, interestingly, this wasn't always a primary target, but with the rise of antibiotic resistance to other drugs, targeting the membrane is becoming increasingly important.
So if I'm getting this right, we have antimicrobials that target the outer structure. We have ones that target protein production. We have ones that target the genetic code. And then we have ones that target the cell's kind of crucial barrier, like the membrane.
Exactly, yeah.
So it's a really diverse set of attack methods.
Yeah, absolutely. And these four mechanisms, they're really the main ways that we currently combat microbial infections. And so understanding these different approaches, it's absolutely crucial for using current therapies effectively and for developing new ones to overcome, that big challenge of antimicrobial resistance.
So considering these diverse attack strategies, here's a final thought for you. What might be some of the fundamental challenges in developing a single cure-all antimicrobial agent that could effectively target all these different essential microbial processes across all types of microbes, bacteria, viruses, fungi, everything? I mean, it really makes you consider the incredible complexity of microbial life and the continuous need for, research and innovation in this field. Thanks for going deep with us on this.
Yeah, absolutely. Thanks for having me.
Okay, that's it for the deep dive.
Bye.
See you next time.